What I'm announcing here is the Vertebrate Genomes Project, organized by the Genome 10K Consortium.
And I just want to set the scene a little bit to think about why today is so extraordinary. So this year marks the 75 years since Ernst Schrödinger wrote his book, What is Life? And in that little book, he mused about what was the raw material of inheritance, what potentially was DNA. And he's a physicist who moved into biology to think, well, it had to be some form of aperiodic crystal, and there had to be a way to try and maintain this crystal, but yet they didn't know how it was replicated. And this little book spawned a lot of the ideas of Watson and Crick and Rosalind Franklin and Mark Wilkins, and the scientific consciousness moved towards discovering what DNA actually was. But that was only 65 years ago. And there are people alive today here in this audience who were born earlier than that, I am sure. Not me, of course. But think about it. 65 years ago, they didn't know really, was it the nucleotides, was the protein? What was the raw material of inheritance? And now, today, we're able to sequence the entire genome from multiple different species, like this weird Sicilian. I know Mark Wilkinson won't agree with me, but we're now able to go and look at all of life. We're able to uncover the raw material of inheritance. We're able to then use that information to make trees of life. And now with these new exquisite genomes, 15 we're talking about today, we're able to uncover maybe what are the genetic bases of the rare, weird adaptations that these extraordinary animals actually have. And so this is a landmark event, because in 65 years we've come along so much. So in 2009, when Steve asked me to come to Santa Cruz to talk about the idea of Genome 10K, which really was quite outrageous, because we didn't have the methodology yet, we didn't have the DNA, we didn't know how we were going to sequence it, but Illumina had come on the scene, and now it was possible maybe to sequence these genomes. Within a decade, we realized that we were going to have to get much better types of DNA. We had to try and work out how could we assemble and move things forward. And as a group, we brought together mathematicians, geneticists, conservation biologists. We brought together zoologists, people who actually know these animals, where they exist, what's their unique lifestyle, and also with access to the DNA. And really, G10K spawned VGP, spawned BAT1K, spawned the Earth Biogenome Project that Harris is going to talk about. And that today we're now able to uncover really the blueprint of all different species. We're able to look at each different base pair, potentially assign it to some type of functional aspect. It's unique. And for me, as part of BAT1K, the idea that we could actually now go and sequence the genome to that level of exquisite definition of every living bat on this planet, which I think everybody should be working on personally. They're the most extraordinary mammals. We're going to be able to uncover what is the genetic basis for flight, for these long, healthy lifespans, for some type of immunity against different viruses such as Ebola. They don't die from Ebola. They don't get cancer, and it's all in their genomes. And now we're going to be able to uncover it. But it took a decade from 2009 till now to get to where we are. But I think that you have to, for good things come for people that wait. And we move the field forward, and this is just a start. And the floodgates will be opened. And why should we not now sequence all of life to see how it's evolved on this planet? And so I'm very glad and honored to be here, and we can talk about bats anytime.
Bats and all other vertebrates. The Vertebrate Genomes Project has set out to sequence and assemble the genomes of all vertebrates at high quality. These are to be reference-grade genomes. For plenty of vertebrates, no genome sequence exists at all. Existing genomes are often incomplete, or there are errors. This has been true, for example, for the zebrafinch genome. Here are the first 15 genomes from the Vertebrate Genomes Project. 14 animals, 15 genomes, because this group includes both the female and male zebrafinch. There are an estimated 66,000 vertebrate species. There may be 71,000 or more. Tallies vary. Which is why the 150 scientists in over 50 institutions in 12 countries who are part of the Vertebrate Genomes Project have their work cut out for them as they sequence and assemble these genomes. At a meeting at Rockefeller University in New York City, they presented the first 15 genomes and talked about what's next.
We decided we're going to do what's called a reference vertebrate genomes project. Not just 10,000 species, we initially started with the G10K, but all 66,000 vertebrates at high-quality genome assemblies, starting with all orders of vertebrates, roughly 260, all families, 1,000 of them, eventually genera, and eventually all species. And first phase would be our proof of principle project, which we're announcing today. The second phase, I call it the rocking and rolling phase, all right, where if we're starting to do thousands of genomes, then we're really in business. And the third phase, if we complete roughly 10,000 genera, we reach the G10K milestone. And finally, eventually all species associated with other consortiums like the Bird 10K group or the Bat 1K group, also represented here today. So the approach we're using for phase one, all right, is to have these long reads, packed bio long reads in particular, with 10x genomics, bio-nanogenomics, and ARIMA genomics, scaffolding approaches to pull those, what we call these contigs together into the scaffolds using longer and longer and longer range information. And to give you an analogy, a short read is like the height of a man or a woman, all right? Whereas a long read is like the height of the Empire State Building or even bigger, all right? Whereas the scaffolding approaches are like trying to take these Empire State Buildings together and bridge a continuous set of buildings between Earth and the moon, right? And what does that gain you? An example is shown here as to what these differences or these longer reads gain you is that if you're trying to assemble a genome that has lots of pieces and all your pieces are small, in the case of this, and I think you're kind of guessing what animal it is, but it looks a little strange. This wing here is in the wrong place. The face is turned around. It's inverted. There are all these gaps in here that can have some errors in them. Long reads makes it easier to assemble. And then with long scaffolding approaches, you can get all the chromosomes together. Now, this would be a perfect genome. We're not there yet, but we're getting much closer. And here is an approach Rong Rei and others in our group have been working on to take ever longer and longer pieces of DNA to string these pieces of DNA into longer and longer pieces until you get chromosomes. And then finally, we'll annotate that is named the individual genes.
Here next to me, Steve O'Brien, Oliver Ryder, and I had the idea that we need to organize the communities that were sequencing various genomes. This was almost 10 years ago. To work together towards a shared technology and an organized exchange of information and mutual support. We created an organization called Genome 10K. The thing that really strikes me is back then, we had no idea how long it would take to get to the point where you had a genome that was complete enough to do science on and could be obtained at a reasonable price. Many of you seen the curves of how the cost of sequencing is going down. But what became very clear, and Eric explained so well, is that what we thought was a genome back then really wasn't a genome that was suitable for doing science. And so it took a long time. Two assemblathons, I see Benedict Payton in the audience, and the Alinathon that Benedict put together really revealed the state of technology then and revealed the fact that it was not adequate to go forward. So we have been keeping the troops excited about this project to the best we can while furiously trying to improve technology along with many others across the planet. I think we've made a turning point at this point. These new technologies for long-read sequencing and long association over a megabase associated one region with the other are spectacular. And we're thinking of genomes now in terms of haploids. We're talking about reconstructing both chromosomes separately, which is a completely alien concept in the old days, but that is what it takes to get it right at this point.
The advances in this long-read sequencing and long-range scaffolding technologies is revolutionizing de novo sequencing. I mean, I basically left the field in 2003, 2004, because with the short reads, I just wasn't, basically producing really high quality genomes was off the table. I mean, I saw that, and I didn't, as an informatician, didn't want to play. That wasn't my game. But when these new long-reach technologies came in, about a half dozen years really started to come into their own, I saw the potential, and I think several of my colleagues saw the potential for us to finally be able to produce gold standard genomes, things that are as good as the best genomes we have today. I mean, you can basically, out of the box now with these technologies, produce a genome that's as good as the Drosophila genome, which is the best sequenced model organism, release 6. You can get the same numbers of continuity and statistics out of this. So the 15 genomes that we're talking about today, okay, I mean, people have talked about, we're going to do 1,000, we're going to do 10,000 human beings, blah, blah, blah. We're talking about, forgive me, low quality stuff, okay? What we're talking about are things that will basically, the word was bandied about with the Human Genome Project, things that will stand the test of time. These are things, it's going to be one and done. We're going to do these things, and we're not going to have to do them again. Almost everything else that's been done to date is going to have to be done again if you really want to do your science. So I think that, I mean, what's exciting about today is what this is it's only 15, but it's the first 15 of what's going to be a flood. So what's happening today and what we're announcing is the beginning of a trend that's basically going to completely and dramatically alter genomics in the future. Thank you.
So I wanted to say, I mean, people have said how this is transformative. It's essentially 100 times longer contiguity than the vast majority of genomes currently in the archive. So that's a good number to keep in your head. It's not alone. There are other genomes out there, like Drosophila and human and a few others. But the 15 genomes we're presenting are, in fact, a major contribution to that set of things at this level of millions of bases rather than tens of thousands of bases of contiguity. And despite 20 years of work and all the new technologies, there still are great technical challenges in piecing together the billions of letters in a genome in the right order with the goal of no error. And in particular, in scaling that up so that we can do it at the level of hundreds and thousands and tens of thousands that we want to. So it's great to be part of this team. It's great to be back working with Jean, who we worked together 20 years ago, and David and others here, and have new people join. And the team here, in my view, is leading the world in attacking these problems, and it's an exciting time in the field. So. I want to, it's interesting conversation, and my combative, contradictory tendencies have been raised. So I think absolutely the goal has to be to get it correct. I actually think, though, you can't let total perfection get in the way of, there is a balance between perfection and utility, which we have to think about and address and meet on each occasion. So,
but I wouldn't agree with that. I would agree with that.
Yeah. So anyway, and the other thing that's really important is actually the speed. The speed is important because what happens with technology is it's increasing faster than twofold a year, and computing technology increases slower than twofold a year. Mm-hmm. And over the last 30 years, we have managed to keep the computing to sequencing costs balanced by improving the efficiency of the computation. And that has been done through a whole lot of algorithmic invention and insight. At times, I think we've been at the cutting edge of computational algorithms in this field, which is kind of surprising.
I think we are at the cutting edge.
And secondly, but also we've taken advantage of the fact that the more we sequence, the more it looks like things we've seen before, and there's a lot of structure there. So I think Jean is right. We have to absolutely get it right. We have to understand what right looks like and be getting it right. But then there is going to be a lot of scope, and it's going to be necessary to make that time and space efficient, compute efficient. Right. And we're not going to manage to scale up 1,000-fold, which is what we will do. We need to do and will do without becoming yet more compute efficient than we are now. So I think that's fun. And then the second thing I wanted to say is that...
Can I respond to that?
In a second, but I'm going to have a second thing too. The second thing I wanted to say is I think that this is kind of a key thing is the relationship between sequences. So there's a relationship between these pieces we have and how they relate to the truth of the individual genomes, but then the relationship between the individual genomes. And we're building a reference resource, and our concept of reference is in a state of change at the moment. And people have, in fact, and David and I have talked about this for years, people have kind of known it's necessary for that to happen. And at the computational level, we've talked about graph genomes, and they haven't really taken off. And I've come to realize, and I think that's still a really essential thing. In fact, the data structures used, some of which Gene introduced for assembly, are very closely related to the ones we need for representing genetic variation within and between species. And so we've all published on forms of this over the last decades. And I think that that's a really important area still. But I've come to realize that you don't really want to expose all that, just as we don't expose Burrows-Wheeler transforms and fancy hash tables to end users and finite state automata in BLAST. That wasn't what the BLAST user knew about. We don't want to tell the end user that our reference is a graph. We want to give them something that lets them look at any individual genome in the context of all the other things related to it and transfer information between them. So we have to work out the modality for presenting that to people and what to put under the hood for doing that. And I think that remains a major challenge. After several years of effort in conjunction with people in Santa Cruz, we've just published in Nature Biotechnology this VG package, which is a piece of that, from my perspective. So I still see that whole agenda as open. There's a huge amount to be done methodologically and computationally in getting the right assemblies and in relating everything to each other and in making it all scale so that we can...
So I agree there. Yeah, so I mean, Richard and I have known each other for a long time, and actually one of the reasons I like working with Richard is because, I mean, as a mathematician, I do tend to suffer from perfectionistic tendencies. So it's wonderful that Richard offsets me with a certain kind of...
English pragmatism.
English pragmatism. I wasn't going to put English in there, but since you did, I guess it's okay. I mean, really, the point, I actually agree with everything that Richard has said. I mean, I only stated what I stated because I feel that much of the work that I've seen has placed the emphasis on efficiency before the aspect of correctness, right? I mean, you of course you want your processes to run in a speed and with a turnover rate that allows you to basically do your work. In fact, I mean, the assembly of PacBio data was taking 400,000 CPU hours, and I brought it down to 2,000, which was basically, and I would agree, at 400,000 CPU Amazon cloud hours, it's totally untenable. I mean, then you really have an issue. But now that it's down to 2,000 hours on an HPC cluster, we're at least back in the envelope.
It's good for now.
And you're right. We're going to do and there's going to be room for more.
There will be room. That's
and the shorthand of how did you do that? The shorthand of how you did that.
My wizardry.
Well known wizardry.
Wizardry, computer science stuff. It's it was. Yeah, I mean, it's not an easy. I think wizard is the best answer I can give.
But it's not magic.
No, but it's you.
Creative.
I mean, but you have to seed things efficiently. You have to find things that are likely to be similar in more and more efficient ways by index structures, and then you have to have fast ways to evaluate and align around them.
Right, so Richard's correct in the sense that the overarching way to do these comparisons is what you do is you build a pre-computed index. You take each of the objects you want to compare and you build a representation of it, an index, that facilitates the comparison of the objects. And that
And this is,
generally
this is
computer
pretty
science
central to computer
has
science
many
now.
such
Yeah.
techniques.
I mean, the whole of the internet search is all based on not the same but related things.
Massive index.
And in
Right,
fact,
massive index.
you know, there's a lot of areas of connection between what we do and what.
I think we're only beginning to really address these deep problems that we'll have computationally. The assembly is the first line of attack to the raw data that we have coming off the machines. But ultimately, the comparison for scientific understanding between thousands and thousands of genomes will require an enormous additional numbers of efficiencies as the type that Gene is talking about here. That is compounded, I think, by the richer conceptual framework that is required.
You have to get the representation
Which Richard
right.
was referring
Yeah,
to.
and that comes down to what Richard was talking about in terms of the representation. It is not, we see people come from computer science into genomics and want to apply their tricks, but it takes them many, many years before they understand the true depth of genetics. If you look at this, if you look at what we're dealing with an evolutionary process on the scale of billions of years that has gradually molded these genomes of different species into what they are today through a very complex set of processes. It isn't just copy-paste, it isn't just make a substitution here. The actual driving changes are very subtle, things like gene conversion and so forth. When you actually want to model that process, it gets very complicated. One has to make compromises in order to do it efficiently. But ultimately, the fact underlying this, the conceptual picture that I want people to take home, is that actually these bases at the DNA base level derive one from another through this process of evolution. I loved Emma's aperiodic crystal to bring back Schrödinger in here. So the information in that original aperiodic crystal has been copied again and again with modification, but modifications of a very rich type. But when we look at a piece of DNA, when people look at a piece of DNA in the Human Genome Browser and they turn on the comparative genomics tracks, they want to see the relatives of that piece of DNA in other species, but increasingly they also want to retract that to a state that it was in earlier. And we actually, for another piece of our work on Kratzing fingers, so we actually reconstruct the ancestral form of the DNA segment, from that derive the protein, and then actually create the protein in its ancestral form and do experiments on it. So to understand evolution, you have to understand evolution as a historical process for which biomolecules existed 100 million years ago, 1 billion years ago, that we can infer and experiment with, but only if we get the relationship between all of these trillions of bases of DNA reasonably correct. Not perfect.
The more perfect, the better.
Yes, but I think we're talking about enough to do science. I liked Eric's introduction because what we had before was not enough to do his science, and that's the true motivation for digging deeper in the technology. We don't know exactly what the formula is that's the minimum to do science, but I'll tell you what, we want to emphasize what Gene was saying. When we tried to compare the genomes that were only partially assembled in little pieces to each other and reconstruct their evolutionary history, it was not enough to do that science. We did not have enough information in that sequence, and it's going to be revolutionary to write these new algorithms now that can actually reconstruct the evolutionary history of all of the bases in a large collection of genomes back to their common ancestral form. That's a huge challenge. It's one of the most exciting pieces of software one could write, and we really now only had the opportunity to do it successfully because of these fantastic new genomes.
So actually I should say, we tend to say this is all the first time this is happening, but actually for bacteria, one of the three, what are they called, kingdoms or something of life?
Kingdoms.
Yeah, this has been done. You know, there are hundreds, I think there are thousands of complete end-to-end perfect bacterial genome sequences that have been assembled over the last, particularly over the last 5 years, but over the last, starting 20 years ago. Because there are only a few megabases, and now these, in fact, the long-read technologies before we've started applying them on this scale have been applied to make end-to-end perfect bacterial genomes. And there, it exemplifies, we have seen that evolution, absolutely critical things in evolution have taken place through structural variation and insertions and pieces which are distributed. Now people can say the mutation process and the recombination and DNA exchange process is different in bacteria. It is to some extent, but I think there's more of what goes on in bacteria, recombination, that's also happening in humans, in eukaryotes than people have thought in the past. So now a lot of what we're interested in life is not bacterial, so there's a huge amount to be done. It's fine.
Well, the structure of eukaryotic genomes are so much richer too.
They're just the whole of life is rich. If we just, if life was all bacterial, life would be the poorer for it. So, so I just want to say, there is material there to work on at one level with respect to all these things. The thing I want to say is, we can talk about the relationship between things, which is evolutionary, which is that they've descended by mutation and recombination and drift and selection. So these are population genetic concepts, and one of the great things is that there is a mathematical structure. Even if it's more complicated than just single bases changing from A to G and so on, which is absolutely true and necessary, it's a relatively, the set of basic operations out of which DNA sequences are made can be reduced into a small set. And there is a theory, and it's a rich theory, which has developed over the last, getting on now for 100 years. We're coming up to almost the centenary of people beginning to write down these processes and think about them mathematically and analytically. And these would be, the holy trio in that case is Ronald Fisher, Sewell Wright, and JBS Haldane, and their descendants. And for a long time they worked in the abstract, actually. And
they didn't have data.
And now they had limited data. They did amazing things with limited data, but now it's all laid out. And it's that which underlies the theory. We're putting together theory, computer science, and data, and biology, and it's an interesting time. And that gave rise, it was a major component of the origin of statistical theory, came out of those processes. So Fisher, in particular, is seen by some as a possibly slightly wayward father of both population genetics and statistics, modern statistics. So I think this issue about how sequences are generated and converted from each other and how they relate to each other and how you efficiently manage and compute and make inferences about things is an incredibly rich area.
Fabulous. I'm keeping this.